So my talk, since I was told this is an education course, so I did thought about what would be the goal. And for those, I see a lot of really first-class researchers in the world and on this electrophysiological source imaging problems. I apologize to the active researchers. I did make a lot of slides for students, okay, for newcomers. Try to share some very basics. So if you see something that you get bored, please forgive me. But I do have some recent results, hope to serve two purposes. So with that, and so the whole major goal for this talk is because this entire education course of the EEG and MEG connectivity. And I assume most of them, if not all of them, is a lot of them, as Thomas in his introduction already says, solve the inverse problem, look at the connectivity in the source domain. So I've tried to provide an overview or some basic coverage on how to solve the inverse problem. What are the challenges? What are the key issues if you want to solve the inverse problem, bearing in mind to try to get the functional connectivity. So this slide just a general background to illustration and to repeat some of the things in the previous two speakers that our brain activity is a spatial temporal process. If you're going to look at the spatial resolution or spatial distribution, then probably functional MRI is going to give you very good spatial resolution. But if you want to see the dynamics, as Christoph just showed beautifully, if you look at the temporal dynamics, and that fMRI is not there yet. Or maybe some technology someone's going to invent in the future, but at this time, EEG and MEG remain to be the only non-invasive human brain imaging technology that allow us to track the temporal dynamics. What we try to do is get both temporal dynamics and the spatial detail to a degree that is allowed by the physical principle. So this is a summary. Probably many of you have seen in various review papers. This is just taken from one of the review papers I wrote several years ago. And just to summarize that, if you look at the hemodynamic or metabolic and imaging modality, which give you the spatial resolution, that is, as what we just mentioned, it's not there yet. The EEG and MEG, if you don't solve the inverse problem, it does not give you spatial resolution really we would like, because there are commercially available high-density EEGs, 256 electrode, MEG device give you 300 plus something channel. That would be the spatial resolution. But if you solve the ESI, or electrophysiological source imaging problem, that is a topic of this presentation, that can bring you down to a much greater spatial resolution while enjoy the high temporal resolution. Now, certainly you could integrate with functional MRI to do multimodal imaging, but that is not topic of today's presentation. There are two other modality, which is shown by this, this would work. Okay, these two guys, these are single unit activity, multi-unit activity, local field potential. These is going to give you high temporal, high spatial, but unfortunately, you have to put the many electrodes into somebody's brain, which is certainly doable in animal. That's a lot of neuroscience research topics, but clearly for obvious reason, that is not an option for human brain mapping, and except for a very small limited case for neurosurgical and patient. So this is just to illustrate about the electrophysiological source imaging and this approach and what kind of technical issues associated with that. And if we want to get to the functional connectivity, first of all, you start from this sensor domain. You get an EEG and MEG recording, so you get the electrical or magnetic functional information that reflect the neural excitation through the volume conduction. At the same time, I do want to mention that even though the community say, our community believe, myself included, that the EEG and MEG really provide functional information, but the good spatial resolution we are enjoying today, to a large degree, is because the greater success of MR technology, because the T1 weighted MRI does provide very detailed anatomical information which are served as something called anatomical a prior in most of the inverse source solutions, which allow us to derive good spatial resolution from EEG or MEG. So by combining these anatomical information with electromagnetic function information, you can do source imaging or connectivity imaging. Either you do the cortical surface, or you could do the volume source imaging, or even one more step from there, you could derive the functional connectivity using whatever the functional and connectivity algorithm. So here is just to illustrate a little bit further in what I mean, because in my title I talk about solve the inverse problem. And if you have the source, you have volume conductor, and then you calculate EEG or MEG, this is usually called the forward problem. But if you get a experimental or clinical recording of EEG or MEG, you solve the inverse problem with the knowledge about the volume conductor model. That is so-called the inverse problem. This inverse problem is going to give you the source distribution inside the brain. From there, you could obtain the functional connectivity further. So that is mainly the topic of this workshop, and that will be my presentation as well. So I was thinking that if for a graduate student or postdoc try to start learning some basics about this problem, what would be the key issue? So here are several key issues I consider are important from educational perspective, which include high density recording, neural source modeling, volume conductor modeling, and source imaging algorithm, connectivity imaging algorithm, experimental validation to make sure, as Thomas mentioned, something works doesn't mean it's true. You have to make sure that whatever your algorithm does give you true solution, you need to validate. Finally is neuroscience application or clinical application. So I want to make a claim that it's not inclusive at all. I made this slide this morning, and I got up at 6 o'clock, and I tried to do a final touch on my slide. So I just went to the Google website, grabbed some picture available. I know there are many other companies available these days, but it does give you a basic flavor that you either high-density EEG recording or high-density MEG recording. Now, the good thing, I think the success of MEG community is virtually all the MEG device, because of the cost, of course, with a price of cost, and they are all high-density, okay? For the EEG at the same time, which is very cheap, and because my lab, and we just added one of the many, and 64 channel and a mapping system, I try to stay away from the company name. I was told, my student told me it's only $40,000. So you can think it's very cheap, but you could get 124 for 8 channel, 256 channel. I know Christoph enjoy a lot of that high density mapping system, but others may want to go with slightly lower. But regardless, the high density recording is important. And how many is absolutely needed? I was fortunate to be included in a group of authors writing a guideline for International Federation of Clinical Neurophysiology. Christoph is one of them, and some of them in the audience are co-authors too. And we debated, and I think we are thinking that something like, it's really different people have different opinion. But minimum we are considering 64 channel is probably the minimum. Hopefully you are going to go 64 to 128 would be the range or consider as high density in terms of EEG. And if you could search for MEG, all of them are already high density. Now, second issue is that source modeling. Now, what is happening in our brain? This is just taking one example about the pyramidal cell and cortical source, cortical neural excitation. And I still recall, the Roberto Pascamacchi, long time ago in one of the workshops I organized, it's 2000 in Chicago, he was explaining his Loretta, that Christoph is a co-author, this famous Loretta work. The explanation that You need to have a synchronized source. And this is one of the arguments I really enjoy a lot. I have been speaking in National Academy workshop, is to say that sometimes the people argue that EEG or MEG does not reflect a single neuron activity. So my argument to this broader community, I told the core audience, I say, yes, if you want to imaging a single neuronal excitation non-invasively, EEG and MEG is not a solution. And even though if there is a solution, I believe there is no solution at this time in this world. Now, what EEG and MEG can do is to imaging the brain function. Because the brain function and dysfunction is encoded in a distributed, synchronized neuronal network behavior. That kind of synchronized neuronal network behavior, when many cells are activated in a synchronized way, that's going to generate detectable EEG or MEG. That's what the EEG MEG is going to tell us. Therefore, the EEG MEG source imaging or electrophysiological source imaging or connectivity imaging is essentially a functional neuroimaging modality. It doesn't tell us function or dysfunction of the brain, although it doesn't tell us a single neuron excitation. If we will remember what we learned in physics course, I don't care what is the Brownian motion of single molecule. I do care the temperature of this room. So if you are interested in the realistic metric, EEG MEG is a solution. If you want to know the very, very detail, then this may not be the solution. So with that kind of comment, and here is say, if we have all these cells are lining up as firing synchronously, we all know that based on general knowledge, we will have current density flowing perpendicular to the local curvature, and if in a global way, using quantitative and imaging science or physics language, we'll see current density distribution located in the three-dimensional volume conductor. And if you put a sensor over the scalp, if it's an electro, you record EEG. If it's an MEG sensor, you record MEG. So that is what is happening. Now there's two issues to solve this problem. One is you need to model the source. The second is you need to model the volume conductor, because you have current density distribution inside the brain due to neuronal excitation, which is going to be impacted by the various inhomogeneity and how easy the current is going to flow through. So you need to model them all together. So some time ago, people thought this is very difficult, but thanks to everybody in this room, and they are not in this room, working over the past decade, we are able at this time to actually to do very good modeling. That is a really major contributor to the success of the current EEG and MEG source imaging problem. So here is a very simple, it's a very basic, oops, I did not realize which is. So you can see, first of all, the source model. Okay, so there's two kinds of source modeling. 20 or 30 years ago, people used to do point source modeling, or for example, most popular one is dipole source localization. Okay, dipole, this is just for those students who are not get used to point source, you're already doing the source imaging, and this is sometimes what we do. And actually, even I wrote my PhD dissertation many years ago using dipole source localization, single dipole, all right? But sometimes it works. Now, recently, there are lots of these distributed source model has been developed and used and successfully established, including two-dimensional cortical source distribution, which could be potential, could be current density, and then three-dimension, which is a current source distribution that is the most popular these days, well accepted by the community. Now, certainly there are two kinds of the volume current source model. One is just to put the entire brain. You just assume that there are all the current dipoles or current source everywhere. Another is assume these are only on the gray matter, excluding the white matter, which you're adding the anatomical constraint one more step further. The volume conductor model, early days, even though now, people still using three spheres model, which is a reasonable approximation of the anatomy and the conductivity and the difference. So three spheres, or some people maybe four spheres, and in MEG usually is a one sphere, which is sufficient. And now if you want to get a high resolution, either MEG or EEG, and then that is this realistic geometry and probably piecewise and homogeneous model is the most popular one. Basically, at least it will include the brain tissue and the bone and the skin. Or sometimes the people put their the fluid and as well. Or if you want to really study special case, then finite element or finite difference and a method could be used. So once again, from the educational purpose, I intentionally keep this slide with equation. I promise that's the only equation I'm going to show today. So this is if you are going to solve the forward problem. Theoretically speaking, one must solve Maxwell equation. But fortunately, or very conveniently, it has been established for decades that quasi-static formulation is good enough for biological electromagnetic problem. In the sense, we only need to solve Poisson equation instead of Maxwell equation if you want to solve the forward problem of EEG or MEG. Now, one thing I do want to bring to your attention, and also with this cartoon, which I made personally actually many, many years ago, the forward problem which actually illustrates the need for inverse problem. If you would have a 3-point source, say 3 cortical areas being activated, if you, through the volume and time conduction, you only see one large area activation over the scalp, which means that this volume conduction essentially is a low spatial path filter. It filters out high frequency information, it filters out lots of useful information, but anyway, that's what happened, why this is the case. You look at the Poisson equation. I'm using EEG as an example, but MEG is similar. What we measure is potential phi. What is the true source is a charge density rho, or could be current density J inside the brain. So you can see the relationship between the source and the field is not a linear relationship. It has a Laplacian operator. That means the second spatial derivative acting upon EEG or MEG is then linearly correlated to the source term. So you can think about that the true source is very sharp and the EEG, MEG measurement is going to be smooth. So this is essentially the challenge we're facing explained by the forward problem. But of course, the beauty is we have all these inverse solutions that we could restore the lost information. So in this is the slide just illustrate what kind of technical challenge or issues if you want to get into this field, or you already get into this field or want to do more. You can see that we talk about source modeling and the volume conductor modeling, and then there's another issue which is a challenge, is you need to solve the inverse problem, and this problem can be solved. And there's a lot of argument that people say there is uncertainty of this, actually for the low frequency Helmholtz equation. And if you want to push it for extreme spatial resolution, you cannot solve it. But if you want to have a constraint with a certain uncertainty, say a few millimeters, then the problem can be solved because the numerous study has shown, and they agree with the experimental and the findings. Now, in terms of source imaging algorithm and how many approaches are there, we talk about, and before, whatever you're going to do, the connectivity imaging, you want to get the source information, assuming if you're going to do connectivity imaging in the source domain. We talk about dipole localization already, and there are cortical potential imaging, and recently I have seen some renewed effort to solve the cortical potential imaging since I worked on this many years ago myself, and this is essentially, it's a downward, actually, how to say, decomposition. You move the scalp and EEG potential, which is potential, going down to the cortical potential. So it is an inverse problem, although it is still a field. It's not a current source distribution, does not directly can be interpreted as a neuronal excitation. And then you're going down the current source density, and I want to mention that this weighted minimum norm approach has been really achieved a greater success, and starting early, like Matty Hamilanian's minimum norm solution, and later on the weighted minimum solution, including the famous Loretta, and now I know there are many other Laura or whatever others, and then, or you just call them normalize the weighted minimum norm, and there are a family of such kind of weighted minimum norm solution, which remain to be very useful and could be solved, could be used. Another approach is beamformer or music type of approach. You know, there Leahy, Morsher, and others, and they report this music approach, or then there's a beamformer approach. Essentially, it's called spatial filter. You scan the entire possible source space and get your solution. Bayesian approach, and which also being pursued by several groups, which is mathematically speaking probably is more beautiful, but in many cases you can see that they are equivalent to others. Very recently, the sparse, and thanks to sparse signal processing in many fields, that approach has been introduced to our field as well. So you can see the sparse method are being used as well. So I try to use the graphics instead of equation. This is actually, this result I took from one of my own early paper published in 1987. This is amazing. I cannot believe that how many years I've been working on this problem. And you can guess how old I am. So this is taken from my PhD dissertation at that time, and we proposed to use boundary element model, although it's homogeneous, but it's an epileptic patient and interictal spike. So you find the dipole from EEG, and in this case, we use a CT at that time, actually is not MRI because of very early time. And another, as what I mentioned, cortical potential imaging, that you're starting from EEG, and then you get anatomical constraint and go down to cortical and space and get the potential distribution. You can compare, validate with ECOG recording directly. Here, this is just show one of the early paper. You can see the scalp EEG here is the inverse solution over the cortical surface. Here is the same patient, the recorded ECOG recording, and both certainly are normalized, and you can see that the correlation coefficient is about 84% or higher, which give you some, it's not everything, but it does give you something useful. Here show a more recent, showing an epileptic interictal spike, and then we show this cortical potential imaging result actually correlated very well with the region that being resected, and also with the hot spot, and according to the invasive ECOG and the recording. Now, what is the most, as what I mentioned, is this weighted minimum norm type of the current source and the density imaging, either from MEG or from EEG, either from spatial information alone or incorporate temporal information. You can merge these and you solve this problem. Now, I want to show using some example to illustrate using epilepsy, why epilepsy has been heavily used, because it's temporally dynamic. As Christopher mentioned, the temporal dynamics is very well illustrated in epilepsy. If the spatially, for example, there's another way, if you're going to start Alzheimer's disease, and that brain activity probably is very smooth. Now, the question whether EEG and MEG is the best solution need to be thinking very carefully. I don't know if it is or not, but epilepsy is clear, and probably EEG and MEG will be very good solution. So even though my group also work on a lot of interictal spike result, I want to really cite this work by Christophe's group. I consider this a major achievement. The Geneva group has spent almost 10 years, and they started 152 patients in epilepsy with high-density EEG recording. And then what they did is they compare virtually every modality except MEG. Okay, MEG was not considered, but and what is very interesting to find is that they get high-resolution EEG-based source imaging, and result with certain structure MRI showed really the highest and sensitivity and specificity compared with all other imaging modality except MEG, which was not included in the study. And this is a large cohort of the epileptic patient, which really I think is illustrate that what kind of clinical utility this approach could be. Now, what I do want to touch on is that even though the work has been established beautifully that we could image the interictal source from high-density EEG, as far as epilepsy is concerned, our ultimate goal is to imaging seizure source, because we want to cure the patient to make it seizure-free. So My own group has been working with this problem with one particular technology approach, which we are quite happy with. And in this case is we introduce one specific application of ICA. And by first of all, decompose and EEG data into multiple IC, and then we introduce something called recombination approach to get a single spatial temporal dynamic source map from this approach. And here just shows some of the results. You can see that this is frontal epilepsy. The yellow color shows the non-invasive EEG source imaging of the seizure, and the green shows the resection as quantified by post-op and MRI. Here is a temporal lobe, and these patients are all seizure-free after 1 year follow-up, which illustrates that the usefulness of this particular approach. Here is a comparison to the intracranial and the direct recordings. And here is this one approach is, and you could certainly using the weighted minimum norm such as the Loretta directly to do the source imaging during the seizure. And this is what we compare, and we found that, and by taking into temporal dynamics into this seizure imaging, actually our result, which is illustrated in this case by this brown color, and while you're directly applying Loretta onto whatever the time and signals at the peak during the seizure onset, we found that actually there's a very significant reduction in localization error and of this spatial temporal approach compared with the direct application of Loretta. And here is a very recent result, actually in a group of patients, and we compare our result. First of all, we compare that with here you can see intracranial recording as a defined seizure onset zone. We found this group average is 1.3 centimeter from our non-invasive imaging result to where this invasive recording is going to be. And on the right side, compare with the surgical resection as quantified by the MRI, and we found that a 6 millimeter localization error from the source imaging result to the edge of resected area, which is a quite encouraging and result, and this work is still ongoing. And then I want to mention that some of this sparse source imaging has been introduced and by various groups, and our group included. Essentially is if you try to take advantage of the signal processing, and one particular recent work that what we did is I see from time to time people working in different imaging community, they say, okay, EEG and MEG is beautiful. But you guys cannot get extent. In the sense, you can only get a center of gravity. On the other hand, functional MRI, which I agree. I work on functional MRI too, by the way. So from functional MRI, I can get the extent, right, activated thing, so which is beautiful. So we try to tackle this challenge. The problem statement is, can we estimate the source extent objectively from EEG or MEG? And this is a work we just published last year. In this case, we incorporated the sparse, and the idea is very simple. I just want to explain about 20 seconds here. So here is a, say, true source, okay? First of all, we solve, for example, using minimum norm, or Loretta, or whatever. I'm using very initial, usually it will give you a much larger, as what we all know. And then we introduce a penalty. The penalty is anything outside of this initial solution is going to have a heavy penalty. But within this area is going to have small penalty. Okay, that takes care of the background. That's number one. Number two, we introduce some concept called edge sparsity, which is exactly derived from focal epilepsy source. That is that if I have a square function here, you take a derivative, you could see it here, like this. So there is edge sparse. This is the minimum norm solution. This is L1 norm solution. L1 norm is going to give you 2 spots, and the minimum norm will give you a large area. Now I'm estimating this edge, because at the edge there is a sparsity. And from there, and actually we could get very reasonable estimation using this approach. Here is a computer simulation result that small, medium, and large area extent simulated. and we run many, many Monte Carlo simulation. Here is our non-invasive imaging result. We did not use any a priori information. There's no thresholding. 2 minutes. Yes, I will be finished in 2 minutes. And which show it works. Here is inpatient. We're testing epileptic patient once again. Here is our inverse solution, and these are ECOG recording. These are seizure onset zone identified. You can, this is resection, and we found that these agree as well. So really, we are still, while we are trying to testing much larger number of patients, but our initial experimental result and the systematic simulation result suggests that EEG or MEG can be used to objectively estimate not just where the source originates, but also the extent. And this will be very important if we establish this modality that could make our EEG and MEG modality much closer to other neuroimaging modality and the many, many other people are using. So now I know that many other distinguished speakers are going to talk about the connectivity. I want to mention what kind of the challenge or key issues when you deal with that. And there are two kinds of these functional source imaging which involve with their inverse activity imaging. One is the source location is fixed. And in another work that we just did is that you could make these nodes actually be dynamic depending on your inverse source imaging, and then perform the connectivity imaging. So there is a collaborative work with Fabio Babiloni and Laura Astolfi, and for many years we collaborate along this line. Is you can see that from the scalp EEG data, you can reduce that to the waveform or region of interest in the cortex. In this case, everything is fixed. And then you do apply the Granger causality to either get information flow, or you get outflow, or get inflow. And this approach, we have applied that to epilepsy problem and found it quite useful. And you can see, first of all, we do a beamforming, the scanning, and then depending on that result, we get 3 possible source candidates. We perform Granger causality. We found that this guy and this guy is a true seizure source, and this is not, and which had 100% success rate in 20 and seizure that we started. And by the way, just for some of the students or any colleagues interested in, we developed a MATLAB-based source code in collaboration with Fabio and Laura, and this is in the public domain. The more than 3,000 download, you can share with others after you download. So every source code is being open. You can use this for temporal, spectral, spatial analysis. You could do their limited minimum, their cortical current density imaging, and you could illustrate this Granger causality analysis based information flow. And so everyone is welcome to download and use this if you're interested in, and we apply this to MEG as well. I will skip here. And this is a recent result that in one patient we record both EEG and MEG simultaneously. And then we found that actually there are 2 regions of interest who apply this Granger causality, and both EEG and MEG show that this region is true the seizure driving force source. And this is another, but Laura is alerting me, so I'm skipping here. So 2 summarizing common. One is that electrophysiological source imaging, or ESI, enables non-invasive imaging of dynamic brain activity, leveraging its high temporal resolution and good spatial resolution. The electrophysiological connectome, for some reason I call it E-connectome by learning United States' human connectome project. I try and make it simple, okay? So, you can like it, you don't have to like it, but this E-connectome approach, performing functional connectivity after solving the source imaging problem of EEG and MEG, and promise to, provide additional information of what we are doing. So here is, even though it's educational presentation, some of the original research results are really done and by my either student, postdoc, or former student, former postdoc, and on this group and collaborators, and in this group, and then it was funded by NIH and NSF. Thank you very much.